So if you look back through, the entire history of humanity, across basically every civilization and era, there has always been this one fundamental demographic rule, a pyramid.
Right, the classic population pyramid.
Exactly. You have this massive base of young children and then a tiny, narrow peak of the elderly at the top. But right now, as you are listening to this, we are living through a statistical earthquake.
Yeah, it's completely unprecedented.
It is. For the first time in human history, globally, people over the age of 65 actually outnumber children under the age of 5. So
Yeah.
the pyramid is it's flipping.
It's a profound shift. And for those of you practicing medicine, it pushes us to a massive clinical crossroads. Because while advances in modern care mean our patients are surviving longer, those extra years are often heavily burdened.
Oh, absolutely. The polypharmacy burden alone is staggering.
Right. It's chronic disease, it's frailty, and like you said, polypharmacy. The reality of taking, a handful of pills every single morning just to manage a cascading series of physical breakdowns. So the focus in longevity research is no longer on just increasing lifespan.
Right. It's not just about years alive.
Exactly. The real focus is on health span. We're talking about maximizing the years lived completely free of chronic disease and disability.
And this shifting clinical reality is exactly why we are pulling apart a really landmark paper today for our deep dive. It's Biology of Aging by Rafael de Cabo and David G. LeCouture.
It's a fantastic piece of literature.
It really is. We are using their research to ask a pretty wild question. Is aging an inevitable, unstoppable decline, or is it just a malleable biological process that we can actually hack or, at least slow down?
Well, to understand how we might slow it down, we really have to look at the origins of the process. And evolutionary biology suggests that aging isn't actually genetically programmed into us.
Wait, really? Because it feels so inevitable.
I know, but there is no self-destruct timer coded into human DNA. Evolution simply doesn't care about you once you've successfully reproduced and raised your offspring. The entire biological system is basically driven by early life survival and reproductive success.
So it's not a self-destruct program. It's just evolutionary neglect.
Precisely. And there are two classic evolutionary theories that explain this neglect. The first is mutation accumulation, which is pretty straightforward.
Right, just random errors over time.
Yeah, random genetic errors build up, and because they only really cause problems late in life, evolution just never bothers to weed them out.
That makes sense.
But the second, slightly more complex theory is antagonistic pleiotropy. This means a biological trait might have a dual role. So, for example, a specific gene might evolve because it hyperactivates cell division.
Which is great when you're young.
Exactly. When you are 20, that is an incredible advantage. It helps you grow robustly, heal from injuries quickly, and reproduce. But that exact same gene activity might increase your risk of cancer when you are 80.
Oh, wow. So it's a double-edged sword.
Yeah. And evolution selects for it anyway, because out in the wild, an animal is statistically likely to be eaten by a predator or, die of an infection long before it reaches old age.
I mean, it sounds a lot like planned obsolescence in technology.
That's actually a great way to think about it.
Yeah, like the human body is basically like a smartphone. It's designed to run flawlessly just until the warranty expires, which, in biology is our reproductive age. And after that, evolution completely stops pushing software updates and just lets the operating system crash.
Right. And that concept connects directly to Thomas Kirkwood's disposable soma theory. The soma refers to the non-reproductive cells of your body, right? So your muscles, your organs, your skin.
The stuff we deal with in clinic every day.
Exactly. The theory argues that your body has a finite amount of energy and resources. If an organism spends all its energy perfectly repairing its DNA and maintaining the soma, it just won't have enough energy left to reproduce quickly.
So it's a forced biological trade-off.
Yeah. Evolution forces the body to prioritize maintaining the germ cells, the reproductive code, and it sacrifices the everyday wear and tear on the soma. The body is essentially disposable as long as the genetic code gets passed on.
But if that's a universal rule, there is a massive discrepancy in how different animals actually experience that wear and tear, right? Like, the sources point out the Greenland shark.
Oh, the Greenland shark is fascinating.
They exhibit what is called negligible senescence. I mean, they can live to be nearly 400 years old with almost no obvious biological changes of aging, and their mortality rate doesn't inexplicably spike as time goes on.
Yeah, the Greenland shark is basically a masterclass in slow metabolism and cold water adaptation. But on the total opposite end of the evolutionary spectrum, you have programmed aging in simulparous animals.
Like the Pacific salmon. Right.
Or certain marsupial mice. They reproduce in one massive exhaustive event, and then they undergo rapid biological death almost immediately. The reproductive effort literally consumes them.
It's wild. But humans seem to sit in a really unique evolutionary pocket between those two extremes, don't we? I mean, we live remarkably long beyond our reproductive years.
We do, and evolutionary biologists attribute this to the grandmother effect.
The grandmother effect.
Yeah. In highly social species, like humans and Asian elephants, the survival of infants heavily depends on the care, resource gathering, and knowledge provided by older females.
Oh, that makes total sense.
Right. So in our specific lineage, evolution actually did select for extended post-reproductive survival because having a living grandmother directly ensured the survival of the offspring.
Okay, so if evolution largely stops maintaining our biological software after we reproduce, how does the hardware actually break down? Biologists have identified this specific set of molecular hallmarks where the rust starts to show. Where does that breakdown usually begin?
Well, it begins right in the blueprints. Genomic instability. Every single day, your DNA is bombarded by exogenous stressors like UV radiation and endogenous ones like oxidative stress from your own metabolism.
So just existing causes damage.
Pretty much. Over time, genetic lesions accumulate. We see point mutations, translocations. Functionally, this means you are getting typos in the genetic code or, entire paragraphs of instructions getting pasted into the wrong chapter of a cell's manual.
And it isn't just the code getting scrambled. The physical structure of the chromosomes actually starts degrading too. This is telomere attrition.
Yeah, telomere attrition is a big one. Telomeres are the repeating sequences of protective DNA at the very ends of your linear chromosomes.
Like the plastic tips on shoelaces.
Exactly.
Yeah.
But every time a cell divides, the cellular machinery that copies the DNA, the polymerase, cannot physically copy the very tip of the chromosome. So with every division, the telomere gets slightly shorter.
I always think of telomeres like a prepaid punch card for a coffee shop. Every time the cell divides, a hole gets punched in the card. Once you're completely out of punches, the cell simply cannot divide safely anymore.
And biologists call that absolute limit of cellular division the Hayflick limit. It was named after Leonard Hayflick, who discovered it back in the 1960s. When a cell accumulates too much DNA damage, or its telomeres get too short, when that punch card runs out, an internal alarm system triggers it to permanently halt division.
Which is actually a good thing initially, right? It's an anti-cancer defense mechanism.
Exactly. If a highly damaged cell kept dividing, it would form a tumor, so it becomes a senescent cell.
They stop dividing to protect the body, but they don't actually die and get cleared away. They just sit there. I've heard them described as zombie cells, but functionally, they act more like squatter cells.
Squatter cells, yeah, I like that analogy.
They completely stop paying rent, meaning they stop doing their designated biological job for the tissue, whether that's acting as a skin cell or a liver cell, but they absolutely refuse to be evicted. And worse, they start throwing toxic chemical parties that ruin the neighborhood for all the healthy cells around them.
And that chemical party is a heavily documented biological phenomenon called the SASP, the senescence-associated secretory phenotype.
This ASAP, yeah.
These squatter cells continuously secrete a highly toxic cocktail of inflammatory cytokines, things we see in the lab all the time, like interleukin-6 and TNF-alpha.
Which leads to what they call inflammaging.
Right, exactly. It creates a state of chronic, low-grade systemic inflammation. Inflammaging is a primary driver in the pathogenesis of age-related frailty, arthritis, atherosclerosis.
So the tissue isn't just getting older, it's actively being degraded by its own dormant cells.
Right. And while that inflammatory chaos is happening outside the cells, things are breaking down inside the remaining healthy cells too, particularly with how they handle waste. This is the loss of proteostasis.
Proteostasis, that's just the maintenance of all the proteins in your cells, correct?
Yes. Proteins do almost all the functional work in your body, but they have to be folded into very specific 3D shapes to work. Over time, proteins get damaged or misfolded.
And the cell normally cleans that up.
A healthy cell has a complex trash disposal system, primarily the autophagy lysosomal system, that identifies these damaged proteins, sequesters them, and breaks them down to be recycled. But with age, this autophagy machinery begins to fail. The garbage trucks just stop running on time.
So when damaged proteins aren't cleared out, they start clumping together into physical aggregates. If you zoom out and look at neurodegenerative diseases, this mechanism explains so much of what we see in clinic.
It really does.
I mean, the Lewy bodies we see in Parkinson's disease or the neurofibrillary tangles in Alzheimer's, Those are the physical manifestations of the loss of proteostasis. It's cellular garbage that just piled up until it choked the neurons.
So we have this fully mapped out horror show. The DNA is accumulating typos, the telomere punch cards are running out, the squatter cells are causing systemic inflammaging, and the cellular trash is piling up in the brain.
It sounds bleak when you list it all out like that.
It does, but this is exactly where the science of aging shifts from observational to interventional. We can actually manipulate these underlying hallmarks. And one of the most powerful levers we have is connected to what we eat.
Or, more specifically, what we don't eat. Because the paper zeroes in on a hallmark called deregulated nutrient sensing.
Yeah, your body relies on highly conserved molecular pathways to constantly monitor how much energy and how many amino acids are coming in. The two heavyweights here are the MTR pathway and the AMPK pathway.
Right, MTOR and AMPK.
When you severely restrict caloric input, you silence these nutrient sensors, and the body reacts defensively. Caloric restriction, reducing caloric intake by 30 to 50% without causing malnutrition, has been shown to extend lifespan across multiple species, from yeast and worms all the way to mice and dogs.
Okay, but restricting calories by 30% for an entire lifetime sounds grueling. Do we actually have real-world human data showing this extends health span enough to be worth the pain?
We actually do have several compelling data points. Observational data from the traditional Okinawan population in Japan showed extreme longevity linked to a lifelong cultural habit of below-average food intake.
Oh, right, the Harahachibu practice.
Exactly. There was also the Biosphere the Second experiment in the early 90s, where participants lived inside an enclosed ecological dome. The agricultural system underperformed, forcing the crew into an unforeseen severe caloric restriction for 2 years.
Wow, an accidental clinical trial?
Pretty much.
Right.
And medical monitoring showed profound improvements in their blood pressure, cholesterol, and fasting insulin levels. But the most rigorous evidence comes from the Calorie clinical trial.
That was the first well-controlled study of caloric restriction in healthy average-weight humans, wasn't it?
It was. The researchers aimed for a 25% reduction in calories over 2 years. But the human participants, struggling with the behavioral reality of just eating less, only achieved an 11.9% reduction.
Honestly, even 12% is tough to maintain daily.
It is. But even at that moderate level, the biomarkers were stunning. Core markers of biological aging decreased, their liver enzymes improved, their core body temperature dropped slightly, which is a classic hallmark of longevity in animal models, and their baseline inflammation plummeted.
But still, even an 11.9% reduction requires constant vigilance. For most people, the mental load of walking around feeling moderately starved for decades is just a massive hurdle. How can the average patient get the biological benefits of silencing those nutrient sensors without the daily misery of chronic restriction?
Well, this is where periodic fasting, or time-restricted feeding, shows incredible promise. Protocols like the 5-2 diet, where you eat normally for 5 days but heavily restrict on 2 days, or eating all your meals within an 8-hour window.
The classic intermittent fasting.
Exactly. Periodic fasting triggers a profound metabolic switch. When the body depletes its readily available glucose reserves, it is forced to shift to using fatty acids and ketone bodies for fuel.
Which trains the cells to be more resilient.
Yes. In mouse models, intermittent fasting drastically lowers blood pressure and protects against the progression of neurodegenerative diseases.
There's an amazing detail in the sources about fasting and chemotherapy too, which is super relevant for the oncologists listening.
Oh yes, the fasting and chemo data is incredible.
When mice are fasted before receiving toxic chemotherapy drugs, the healthy cells go into this dormant protective state, while the tumor cells, which are biologically wired to constantly demand glucose, remain highly vulnerable.
It's brilliant. The fasting protects the healthy tissue from the side effects, while the chemo destroys the cancer.
It is a fascinating application of metabolic switching, but researchers understand that even intermittent fasting is incredibly difficult for widespread public compliance. So the current holy grail of aging research is finding a way to replicate the cellular effects of fasting pharmacologically.
Right. Tricking the body's nutrient-sensing pathways with a pill.
It's like hacking a smart thermostat in your house. You aren't actually opening the windows to drop the physical temperature, meaning you aren't reducing your actual food intake, but you hold an ice pack to the thermostat sensor. The system thinks the temperature dropped, so it forces the body to turn on the furnace of self-preservation and cellular repair.
That is the exact mechanism of action for a class of drugs known as CR mimetics, caloric restriction mimetics.
Yeah.
You are manipulating the cellular sensors without the behavioral misery.
And the paper highlights four major pharmacological agents here. The first one gained massive public attention because of its association with red wine, right? Resveratrol.
Yes.
So how exactly is a compound from grape skins hacking our cellular thermostat?
So resveratrol activates a family of proteins called sirtuins. Sirtuins are heavily involved in regulating gene expression and DNA repair, but they are dependent on a molecule called NAD+, which fluctuates based on our energy levels.
Okay, so it's directly tied to metabolism.
Exactly. Resveratrol effectively mimics a low-energy state, stimulating sirtuin activity. This suppresses inflammation and improves mitochondrial function, mimicking the gene expression changes we typically only see during actual caloric restriction.
Red wine compounds are interesting, but the next agent is arguably the heaviest hitter in longevity research right now, and it has an incredible origin story, rapamycin.
Rapamycin is fascinating. It was discovered back in 1964 in a soil sample taken from Easter Island, which is known indigenously as Rapa Nui.
Which is where the name comes from.
Right. Researchers found a bacterial byproduct in the dirt that acted as a potent antifungal. But then they realized it stopped cells from dividing. Rapamycin acts as a direct, potent inhibitor of the mTOR pathway. Literally, mTOR stands for the mechanistic target of rapamycin.
And because mTOR is the cell's primary nutrient sensor, inhibiting it makes the cell believe it is starving.
Exactly. It immediately turns down protein synthesis and ramps up autophagy to recycle all that cellular trash we talked about earlier. Rapamycin extends lifespan in virtually every single organism tested.
But here's the clinical catch. If rapamycin is the ultimate mTOR inhibitor, why aren't we all taking it right now? There is a massive clinical hurdle here. We already use it in medicine, right? To suppress the immune system of organ transplant patients so they don't reject the new organ.
We do.
We obviously can't have the entire population walking around immunocompromised just to slow down aging.
Right. The clinical side effects of chronic mTOR inhibition are significant. Daily use can lead to impaired wound healing, hypercholesterolemia, which is a severe dysfunction in lipid metabolism, and proteinuria, where you leak protein into your urine, signaling pretty significant kidney stress.
So how do we get around that?
Well, the challenge is decoupling the longevity benefits from the immunosuppression. Researchers are currently experimenting with intermittent dosing strategies.
Oh, like taking it once a week instead of daily?
Exactly. By giving mice rapamycin periodically rather than daily, they can trigger the autophagy and lifespan benefits while allowing the immune system time to recover between doses.
Okay, so if suppressing mTOR is too risky for the average patient right now, what about attacking the cellular garbage problem directly? This leads to the third mimetic the authors discuss, spermidine.
Spermidine is a polyamine that naturally occurs in our bodies and in foods like aged cheese and mushrooms.
Which is great news for cheese lovers.
Definitely. It naturally declines as we age, but interestingly, human centenarians manage to maintain high levels. Spermidine directly promotes autophagy, effectively bypassing the mTOR pathway to stimulate the cellular cleanup process. Oral administration has actually shown strong cardioprotective effects in animal models.
Which brings us to the final drug, and this one is already sitting in millions of medicine cabinets around the world, metformin.
Oh, metformin. It's a big one-eyed derivative originally sourced from the French lilac plant. And it is a globally prescribed, incredibly cheap drug for type 2 diabetes.
We prescribe this constantly.
Right, it's ubiquitous.
Mm.
Now, while rapamycin targets the mTOR pathway, metformin primarily interacts with the AMPK pathway, the cell's fuel gauge.
So it works on the other side of the metabolic coin.
Yeah. Metformin mildly inhibits mitochondrial complex I. Essentially, it slows down the cell's power plant just enough to reduce toxic exhaust, which is oxidative stress, and artificially lowers the cellular fuel gauge, triggering AMPK to initiate metabolic defense mechanisms.
And the push for metformin in longevity isn't just based on mouse models, right? The observational data in human populations was an absolute wake-up call for researchers.
It was huge. Large retrospective studies revealed a startling trend. Type 2 diabetics taking metformin were not only outliving diabetics on other medications, they were in many cases outliving healthy non-diabetics who weren't on the drug.
Wait, really? Outliving the healthy controls?
Yes. They were also experiencing lower baseline rates of cardiovascular disease and certain cancers, and this realization led to the design of the TANE trial, targeting aging with metformin, spearheaded by Dr. Nir Barzilai.
Because the FDA doesn't recognize aging as a curable disease, right?
Exactly. So the TAME trial is enrolling 3,000 human subjects between the ages of 65 and 79 to definitively see if metformin can delay the onset of a whole cluster of age-related diseases simultaneously.
That could completely change our prescribing habits.
I got that.
Okay, so we have the nutritional lever of fasting and the pharmacological lever of CR mimetics like metformin and rapamycin. But there is one completely free behavioral intervention that reveals a massive biological paradox: exercise.
The ultimate intervention.
Right. We know exercise improves health span and prevents sarcopenia or muscle wasting. But if aging is essentially the accumulation of damage over time, the DNA typos, the frayed telomeres, the oxidative wear and tear, why does intentionally inflicting physical stress and microdamage on the body through intense exercise actually protect us from aging?
It's a great question, and you're pointing to a fundamental biological principle called hormesis.
Hermes.
Yeah. The biological concept of hormesis dictates that exposing an organism to low doses of noxious stressors actually preconditions the body to survive massive lethal doses later.
So when you are out for a jog and your muscles are burning and experiencing micro tears, you aren't just burning calories, you are literally forcing your cells to run a biological fire drill.
Exactly. When you undergo the physical stress of exercise, or, sit in a dangerously hot sauna, or experience a brief lack of oxygen, you activate anti-stress transcription factors like NRF2 and FOXO.
Like cellular construction foreman.
Yeah, that's perfect. The mild stress wakes them up, and they immediately order the production of powerful antioxidants and heat shock proteins to armor the cell.
Right.
A great clinical example that you guys see in the OR is ischemic preconditioning.
Oh, sure.
If a surgeon temporarily cuts off blood flow to a tissue for a brief controlled period, the tissue panics and mounts a massive adaptive stress response. When the actual surgery happens later, the tissue is fortified and experiences far less damage.
So exercise is just a hormetic stress.
Right. You cause controlled microdamage to trigger a macro repair response, leaving the machinery far more resilient than if you had just stayed perfectly safe on the couch.
Okay, let's synthesize this entire journey for everyone listening. We started with the evolutionary reality that our bodies were never designed to last forever. They were basically just disposable vehicles meant to get our genes to the next generation.
Right, the disposable soma.
We looked at how the hardware rusts, the frayed telomere punch cards, the squatter cells spewing inflammation, and the protein trash piling up in our neurons. And then we explored the emerging toolkit to fight back. We can silence our nutrient centers with fasting. We can hack the cellular thermostat with CR mimetics like rapamycin and metformin. And we can build resilient tissues through the hormetic stress of exercise.
And if we pull all of these threads together, it represents a monumental paradigm shift in how we approach human health. It is the foundation of a field called geroscience.
Yes.
Yes. Historically, modern medicine has played a game of whack-a-mole with chronic disease. A patient has a heart attack, we treat the lipid dysfunction. 10 years later, they develop a tumor, we treat the cancer. 5 years later, they develop dementia, we try to manage the cognitive decline.
It's exactly what we do every day.
But geroscience argues that the biological hallmarks of aging, the senescence, the mitochondrial dysfunction, the genomic instability, are the root cause of all these conditions.
So if you just cure the heart attack without addressing the underlying cellular aging, the decaying biological environment just ensures another disease pops up to take its place.
Precisely. Chronic diseases are simply localized manifestations of systemic aging. If we can target the underlying hallmarks with things like AMPK activators or targeted hormesis, we don't just delay heart disease. We delay the cancer and the dementia at the exact same time.
We started this deep dive talking about a demographic earthquake. We have more older adults on the planet right now than young children. We used to think the fountain of youth was just magic water, but now we are actually reading the molecular blueprints. So here is a final thought for you to mull over as you go about your day or, head into your next shift.
It's a big one.
It is. If the science of geroscience is right, and old age isn't an inevitable, helpless decline, but rather the ultimate pre-disease, a treatable, biological condition that we can manipulate, how should that change the way you view your own timeline? If those extra decades could be lived free of frailty, how does it change your daily habits and ultimately what we expect from the future of medicine?